Hello, hi, my name is Teacher Adib. I'm a science teacher and I teach biology. I have a bachelor degree in science graduated from the National University of Malaysia UKM, and I also have a foundation certificate in life science from University of Malaya. So I'm a teacher who are very passionate about education. I love to learn about life, nature, and science related things myself and teaching them to my students because I want my students to really understand and aware of the wonders that exist around them. I am very familiar with this subject. I've been learning and teaching biology for quite some time and I know how to deliver this subject effectively. So before we start, I would like to take this opportunity to outline my teaching strategy in biology. So in any chapter or topic, I will briefly explain about why you learn this topic, why is it important, and I will give students a clear picture of what they are going to learn. Okay? This is very important because I want to give the whole idea of what it is all about, and I don't want a student to eventually, get lost in the midst of learning. We will establish a good foundation of knowledge of what we are going to learn. I'm open to refresh to students about the basic knowledge and progressively we will explore and gain more knowledge of what we are going to learn. I will also incorporate multimedia approach in my teaching strategy. I might use different tools or multimedia resources such as videos, visual, GIF, and animation so that student can understand better. And the last one, well, I just can't help it. It is my style to make learning fun, interactive, compact, and easy. So I hope you are excited to learn with me. So let's begin our mock lesson. So as for today, I prepared a mock lesson for A-level biology titled Protein Synthesis. Alright students, this is A-level biology lecture. So today we are going to learn about protein synthesis, which is the transcription and translation processes. So in this lesson, you are going to learn about what is protein. I'm going to give a brief introduction about proteins, and then a summary of protein synthesis, the transcription of DNA to form mRNA, and the translation of mRNA into protein polypeptides. Okay, so what is protein? Why protein is important and the significance of protein synthesis? So proteins are organic materials which can be formed, break down, and decompose. Okay, proteins are basic building of human body. A new form of protein can be synthesized in our body. Okay? It can be synthesized by our body cells, okay, which means that we can get proteins from the food or drinks that we consume, and then our digestive system will break down the proteins into amino acids, and these amino acids can produce a new protein which have various types and function to build up tissues, organ, and chemicals in our body. So essential proteins are made by our body cells. Okay, so the important organelles responsible in protein synthesis are the nucleus, okay, the Golgi apparatus and the endoplasmic reticulum, and the ribosomes. So the nucleus contains DNA which store genetic code for protein synthesis, and ribosome is function to translating code from mRNA and making protein, and the endoplasmic reticulum and Golgi apparatus function to modify, sort, and package proteins for secretion out of the cells. So how proteins are made? Okay, this is the summarized flowchart which I want to give the whole idea of how proteins are made. So protein synthesis involves two main processes, which is the transcription and the translation process. Okay, so the DNA in the nucleus will be transcribed into mRNA strand, okay, which store the genetic code for protein synthesis, and then the mRNA strand will then be translated into proteins, into strands of amino acids. Okay. So the transcription process happens in the nucleus while the translation process happens in the cytoplasm with the help of ribosomes. So the summary of protein synthesis, okay, the DNA strand will be transcribed into a single strand of mRNA, and then this mRNA will be translated into
strands of amino acids, okay, which then will be modified into complex protein. All right. Now I'm going to give a brief explanation about transcription process. Okay, so it's all start in the nucleus. Okay, where the nucleus contains chromosome, which consists of strands of DNA, function to store genetic code for protein synthesis. Okay, if you still remember, DNA is deoxyribonucleic acid. It's the genetic material of living organism. Okay, so the DNA have four nucleotide bases or the nitrogenous bases, which known as cytosine, thymine, adenine, and guanine. Okay, and the pair is ATGC. Okay, so in the nucleus, transcription process happen where DNA will undergo transcription process by RNA polymerase to form a single strand of mRNA. All right, this is the general structure of mRNA. So mRNA consists of four nucleotide bases, which is the cytosine C, guanine G, adenine A, and uracil U. Okay, so as in DNA, the genetic code or the nucleotide basis is ATGC. While in mRNA, the genetic code is AUGC, the nucleotide basis. So as you can see, the thymine, the T is replaced by uracil in mRNA. Okay, and then the mRNA will then be translated into proteins, into amino acids and form a complex proteins. There can be up to 20 different amino acids in protein. So this is a summary of transcription process. So the RNA polymerase, will transcribe the DNA strands into a single strand mRNA, and then this mRNA will then leave the nucleus through the nuclear pore into the cytoplasm and then be translated into proteins. All right. Okay, so remember there's two important process, which is the transcription, which happens in the nucleus, and the translation process, which happens in the cytoplasm with the help of tRNA and ribosomes. Okay, so this is the transcription process. You can read this by yourself. Okay, now here comes to the important part of this lecture, which my instruction is very clear. I have to explain about the translation process. So translation is the process of translating the sequence of nucleotide bases of mRNA into sequence of amino acids in a protein. Okay, so the important elements involved in translation process is the messenger RNA, the tRNA, and the ribosomes. Okay, so you have to know what is the structure of mRNA, the structure of tRNA, and the structure of ribosomes. Okay, so mRNA structure. mRNA is a single-strand genetic sequence transcribed from DNA. So it consists of multiple sets of codons. Okay, a codon is a set of 3 bases in RNA sequence that specify a single amino acid. Okay, this one codon will form a single amino acid. Okay, for example, this is 3 nucleotide bases is a set of codon. It's one set of codon. So one codon will produce one amino acid. Okay, for example, uracil, and cytosine, UUC, will produce a specific amino acid known as phenylalanine. Okay, so each triplet codon codes for a specific amino acid. So in mRNA, there are sets of codons. Okay, for example, AUG, AAC, and UCU. Okay, so each codon will be translated into a single amino acid. Okay, now let's look at the structure of tRNA. tRNA has a clover-shaped structure. Okay, however, the most important parts in tRNA is the anticodons, which will be paired with the codon in the strand of amino acid. Okay, for example here, the anticodon is UCA, so it will bind with the codon of ACU in mRNA. Okay, so the function of tRNA is to translate mRNA into amino acid correspond to set of codons of the mRNA. All right, now let's look at the structure of ribosome. Ribosome is consists of a large subunit and a small subunit. So the function of ribosome are to bind with the tRNA hold the mRNA in place during the translation process and assist in the translation of mRNA. Okay, so you have to remember three things, which is the mRNA, the tRNA, and the ribosomes. All right, you have to remember all the structures of these elements. All right, now I'm going to briefly explain about the translation process. Ultimately, translation has three steps, which is the initiation step, elongation step, and termination step. Okay, so the first step is the small subunit of ribosome. Okay, will attach to the mRNA, okay, to the mRNA strand, and then the tRNA will bind with the large subunit of ribosome. Okay, and then the third step is the large subunit of ribosome will eventually attach to the small subunit of ribosome. So it will hold the mRNA in place, and then only translation can begin. Okay. So this is the animation of translation process. Okay. Here we have tRNA, okay, which binds with the ribosome. Okay. So the tRNA will translate the mRNA's, the codon in mRNA strand and produce a specific amino acid. And then this amino acid will be passed to the next tRNA, and then it will form a complex protein. Okay. So how this happen? So inside the ribosome, tRNA which carry a specific amino acid will come in and then binds with the codon in mRNA, and then it will produce a specific amino acid. Okay, after it produces a specific amino acid, it will then leave the ribosome and then comes in another tRNA. All right, and then the translation process continues until it comes to the stop codon, which the translation process will stop, or we call it as a termination stage. Okay. So this is the sequence of amino acids which will form a complex protein, which will form a specific structure and will carry a specific function, and then will be secreted out of the cells. Okay. Okay. Now I'm going to explain details about translation process. This is the mRNA strand, and this is the DNA strand. Okay, as you can see in DNA strand, there are thymine, adenine, cytosine, and guanine. However, in mRNA strand, there are adenine, uracil, guanine, and cytosine. Okay, as you can see, the thymine in DNA is replaced by uracil in mRNA strand. Okay, it's not the same. Okay, so to start the translation process, the tRNA must find the start codon. Okay, for example, here AUG. Okay, so the start codon is the place where tRNA, the anticodons of tRNA, will bind with the codons of mRNA, which is the start codon here, and then only the translation process will begin. Okay, and then it will form a specific amino acid, pass it to the next tRNA, and then the translation process continues until it comes to the stop codon. Okay, for example, UAG or UAA here. Okay, the most important letters is the first two letters, UA. If it's UA, then we know that it is a stop codon. Okay, so tRNA must find the start codon AUG. Okay, so the anticodon of tRNA UAC will bind with AUG and then form a specific amino acid known as methionine. So okay, this is the strand of tRNA, and this is the strand of mRNA. So the anticodons of tRNA will pair with AUG, the start codon of mRNA, and form the first amino acid, which is the methionine. All right, and then the translation continues until the tRNA finds the stop codon ACU here. Okay, and then the translation will stop. Okay, which is the termination process. Okay, so summary for translation process. Okay, summary for the whole process. DNA will be transcribed into mRNA strand, a single strand of mRNA, which consists of 4 nucleotide bases A, G, C, U. Okay, so mRNA is translated by the tRNA into amino acid. Okay, and each codon corresponds to 1 amino acid. It can be up to 20 amino acids in a single form of protein. Okay, and then amino acid will undergo modification and form a polypeptide chain, and this polypeptide chain fall into proteins, a complex protein. Okay, so this is another summary. You have mRNA strand. Okay, this is the tRNA will bind with the codons in the mRNA, and then begin the translation. It must find the start codon, okay, to start the translation process, and then after translation complete, it will form a free complex protein. Okay, so there can be up to 20 amino acids, okay, starting from methionine, for example, and then until it comes to the stop codon, will produce the last amino acid, and this protein will form a polypeptide chain, and this polypeptide chain will then modify into a complex protein. Okay, so remember, there are 2 main processes, which is the transcription in nucleus and the translation in the cytoplasm with the help of tRNA and ribosomes. All right. Okay, that's all for the lecture. Now I'm going to give a quick quiz, okay, of the nucleotide bases in DNA and mRNA. So in DNA, there is adenine will pair with uracil in mRNA. Okay, because the thymine is replaced by uracil in RNA. And then C will pair with G. G will pair with C, cytosine. Thymine will pair with adenine. All right. Okay. And then name the structure below. Okay. So this is the ribosome. Okay, the large subunit of ribosome. And then this single strand is the mRNA. And then this is the large subunit of ribosome, and below it is the small subunit of ribosome. That's all for today. I hope you enjoyed this lesson, and till next time.